Hello, everyone. My name is Matthias Weissensteiner. I'm a bioinformatician and researcher at the Institute of Avian Research. And I'm very honored to be given the opportunity to speak at this event here at the Lab Roots Genetics Week. And today I want to tell you something about sequencing errors and alternative DNA structures. So my talk is structured in roughly 3 parts. In the first part, I will tell you what alternative or non-bDNA structures actually are. In the second part, I'm briefly going to go over the major sequencing technologies and how they work. And in the last part, I will actually talk about a recent study of mine where we have looked at the relationship between non-bDNA and sequencing errors. So to start off with, so to explain what non-bDNA actually is, I first go over the bDNA, and that is the DNA double helix. So this is essentially the textbook representation of DNA. It's a right-winding double helix with 10 base pairs per turn. And the bases are paired in the Watson-Crick base pairing, meaning that A pairs with T and C with G. I only also rather recently learned that this is not the only structure DNA can actually adopt, even in living cells. And there's a variety of different structures, and they're all like commonly called non-bDNA because the canonical DNA structure is called bDNA, and they all so they can adopt different structures, but they depend on a variety of things, mainly the underlying sequence motifs and also a diverse range of chemical and physical conditions. To be more specific here, I will talk more about 6 different types of non-bDNA, which we also have studied in our recent project. And I mentioned already that they depend on the underlying sequence motif. So to start off with bent structures, their underlying sequence motif are A-phased repeats. They're on average about 26 base pairs long, and they're characterized by these adenine tracts, which are structured in a different in a way that they have these A tracts and there's spaces in between. Another non-bDNA structure are slipped strands. So in this non-bDNA, you have direct repeats, so meaning that you have a sequence repeated twice with a arbitrary sequence in between, called the spacer. And when this structure forms, the arms, so repeat 1 and repeat 2, are actually aligning in a shifted pattern, so that the spacer and also part of the arm is sort of looped out, and you have this slipped structure. A rather interesting non-bDNA structure is the G4 quadruplex, and interesting in a particular way because here you have also non-Watson-Crick bearing at play. So in the G4 motifs, you have blocks of guanines, so-called stems, also at least 4 stems in a regular way structured, and they pair with each other. So you don't have the typical C and G pairing. You actually have G's pairing with each other in these Hookestein hydrogen bonds and building this 4-stranded planar structure, which is sort of like a knob. And the other strand can either also form a structure or is essentially just present as a single strand in the cell. Furthermore, we have cruciform structures, which have inverted repeats, so palindromic sequences underlying them. In this case, we also have 2 repeats with 2 repeat arms with a space in between, But in this case, they're oriented towards each other in a reverse complement kind of way. And they're also like intra-strand, so the bases are pairing within the strand. So meaning that you have these strands looped out in a cruciform kind of way. And the spaces then also form this single strand structure at the end. There's also structures that have more than two strands. So before we had the G4 quadruplex, but there's also triple helix structures, also called HDNA. And the underlying sequence motif here are mirror repeats. And there's again two repeat arms, but there's no spacer in between. And they also, as opposed to the inverted repeats, they're just like facing away from each other and are not reverse complement. And then finally, we have another helix structure, but in this case, it's a left winding helix, and as opposed to the right winding double helix, the canonical structure. Here, it's a left winding helix, and it's also structured in a zigzag kind of way, and that's what's causing the name zDNA. And there's pyrimidine motifs underlying this structure. So now you could ask yourself, well, this is very nice, but what are actually like real life consequences or biological implications for that? And so far, there has been a number of studies showing that these structures are actually not just biochemical oddities, but they are also very important for a number and a variety of cellular functions. For example, you have G4s acting as telomere and production. So the telomeric repeats are actually also able to form these G4 quadruplexes, and they may act as a shield against deterioration from nucleases. Again, G4 seem to play a role in the initiation of replication. So this is ongoing research, but the idea here is that the G4 structure actually acts as a docking station, and by that initiates replication. And finally, we also have G4s involved in the regulation of transcription. So essentially, G4s again acting as a motif or a structure that can be recognized by transcription factors. So these are all cellular processes which are essentially vital or like very important for the cell, but there's also examples where these structures can cause issues or even like diseases. For example, G4s are also associated with impeding replication. So essentially preventing the DNA from being replicated, which is obviously not very beneficial for the organism. Or there's cruciforms being involved in genome instability. So whenever these structures are forming in the cell, there is increased DNA cleavage at these structures, at these cruciforms, leading then to increased deletions and translocations. And once again, you can imagine that this has like very severe consequences for the organism carrying this. So finally, for the general part on the non-B motifs, how can these be studied actually? So far I've been talking about their biophysical structure and biological implications, but in fact they can be predicted just with any DNA sequence. So these repeats I've been talking about before, like A phase, direct, inverted, and so forth, or also the zDNA or G4 motifs, they can be predicted computationally. So algorithms have been developed to detect these motifs from just the DNA sequence. And by that, you can get an idea about how common these motifs are. It's important to say here, though, that this is just a theoretical prediction. So meaning that you have a motif potentially forming these structures or any given structure, but it doesn't tell you anything about the like either in vitro or in vivo forming of these structures. So for these, other methods are needed. Okay, so that was the first general part on the non-BDNA structures. And in the next part, I am going to talk a little bit or very briefly about the three major sequencing technologies, which we also have used in our study. So first, to start with one of the most common, or maybe the most common sequencing technology, Illumina short-read sequencing. And here, the basic principle is that a template molecule, so you have extracted DNA from a organism, and this is essentially attached to a flow cell, and adapters have been ligated to it. And then this is amplified with a DNA polymerase to have this clonal amplification step. And then finally, the actual sequencing is sequencing by synthesis, and in which the reverse strand is still present on the flow cell, but via the incorporation of fluorescently labeled nucleotides, the actual sequencing is done. So, and here you can imagine that either in the amplification step or also in the sequencing step, if you have these non-BDNA structures actually forming during the sequencing process, they can sort of interact with the polymerase during the amplification step, and by that introducing additional errors in the clonal amplification step. To go on to the next sequencing technology, Pacific Biosystems. So this is a fundamentally different sequencing type or approach. And in this case, single molecules are sequenced without any amplification step. So it's called single molecule real-time sequencing. And here, template, usually like very long template DNA molecules, so high molecular weight DNA, is guided in these zero-mode waveguides. So these are like nano wells, like really small. And in each of these wells, you have a DNA polymerase and ideally just a single DNA molecule. And then what happens during the sequencing process is that that DNA template molecule gets essentially like synthesized multiple times, or first it gets circularized so that you have these elliptic structure sort of, and then it gets sequenced multiple times over with, again, fluorescently labeled nucleotides. And again here, any secondary structure that is forming in the template molecule might actually impede the DNA polymerase at work here, and by that introducing additional errors or inaccuracies, which are then, of course, influence your sequence that you're working with in the end. Finally, the third technology we have been working with is from Oxford Nanopore Technologies. And once again, this is a very different approach of sequencing. And in this case, there's no polymerases at all applied. And instead, how you get the sequence here is that your template molecule is attached to an adapter. And this, in turn, then is guided towards a nanopore with a motor protein. So this is typically a helicase, which is essentially moving the DNA molecule through this nanopore. And the way you are able to detect the nucleotide sequence is then essentially by very minute changes in the current of this membrane. And by this, you're able to detect then the nucleotide sequence. Also here, if there is a secondary structure forming in the template molecule, this might once again impede not the DNA polymerase in this case, but the helicase. And actually also from living cells, it is known that helicase are essentially applied by the cell to unwind these structures. So, but there is still potential that these secondary structures might actually impede the process of moving the DNA molecule through this nanopore. Okay, so in the third and last part of my talk, I now want to talk about the relationship we have studied between non-B motifs and sequencing errors. And the rather simple question we have asked ourselves is whether in potentially non-B DNA forming motifs, there are more sequencing errors than in a corresponding control sequence. So on the left here, there is the motif with more errors, and on the right, the control with fewer. So that was essentially our starting hypotheses. And for this, we used a publicly available data. So we used the Genome in a Bottle consortium's data of the HG002 individual. And the big advantage of this data set is that the very same individual has been sequenced by multiple different technologies. And again, here we used Illumina PacBio HiFi. So the HiFi mode is a sequencing approach where the consensus of these multiple rounds of subreads is taken, making them very accurate. And the third technology is the Oxford Nanopore. Okay, and then first off, we started by gathering the annotations for the non-B motifs. So before I've shown this NB-MST tool, which is essentially a collection of tools used to annotate any given sequence for these non-B forming motifs. And we have compiled these over 5 million motifs in total, covering about 137 megabases of the genome. So here's just a brief collection of the individual motif types here. And so the zDNA motifs were the least common ones with a bit over 200,000, going all the way to inverted repeats where we found about 3.7 million of them. And so to essentially answer the question posed before, whether there's more errors in the motifs versus the controls, we then also generated control sequences. So for each and every non-B motif type, we took the same number of intervals with the same size, and we excluded any other non-B motifs and of course gaps, but also homopolymers. So any like nucleotide repeated more often than 7 times in a row has also been excluded. So in our data set, then we applied 2 filter levels corresponding to the moderate filter level, where we only filtered out motifs with a mappability of less than 1. And also we applied a size constraint from 10 to 1,000 base pairs. Whereas in the stringent filter level, we were more strict about excluding overlaps with other motifs and also repetitive DNA. So many of these non-B motifs overlap with STRs or simple tandem repeats, so microsatellite repeats, and also repeat mask entries. So for example, transposable elements or other known repetitive DNA. So for the stringent filter level, we have excluded all these. Furthermore, we also excluded any motifs with a with an average base quality of less than 30 or 73, corresponding to Illumina or Hi-Fi base qualities. Okay, and then we went on to actually identify errors in our data set. And as it turns out, it's not that straightforward to find errors, because of course no one actually wants to find them. Everyone wants to make sure that they are working with true biological variants, and everyone wants to exclude errors as much as possible from their data set. So to achieve this, we first took the genome in a bottle true variant set of this very individual to make sure that we're not finding biological variants and only errors. And then for any interval that corresponds to a non-B motif, we essentially counted mismatches where a read showed a mismatch to the reference and it was not overlapping with any true variant sets. So in the given example here, the red square is a non-B motif. And so that makes 3 mismatches divided by 55 nucleotides, an error rate of 0.054, arguably a very high error rate, but this is essentially the approach we have been taking. Okay, so now to the main question, are there more errors in non-B motifs? And the answer is, as so often in biology or in life science, it depends. So first, the Illumina data. In Illumina data, so in this figure on the x-axis, there's these 2 panels corresponding to the moderate and stringent filter levels. And on the x-axis, there's the 6 different motif types. So A-phase repeats, direct repeats, G4 motifs, inverted and mirror repeats, and zDNA motifs. And the red color here corresponds to an increase, fold change of the error rate, the single nucleotide error rate. And blue is a decrease, essentially a fold change of less than 1. So, and what's immediately obvious here is that 2 non-B types, namely G4 motifs and zDNA motifs, seem to show a drastic increase in their error rates. So G4 motifs in the moderate filter set, for example, show over 2 times more increase in error rate than in the control, compared to the control. And this is also still visible in the stringently filtered set. Moving on to the Hi-Fi data, the PacBio Hi-Fi data. Overall, the first impression is that we see a lower difference between controls and motifs. This might be mainly due to the fact that overall there's much fewer errors visible in the Hi-Fi data. But again, there is an increase in the error rate. So there's 1.3 to 1.4 fold more higher error rates in non-B in G4s and zDNA motifs. So this seems to be consistent between those 2 technologies. Finally, we have Oxford Nanopore. And this was quite interesting to us here in the moderate filter level. All but 1 non-B motif actually showed lower error rates in the motifs compared to the controls. And this might be due to the fact that in the motifs we at the beginning excluded any homopolymers except for G4 motifs. And so this might lead to the pattern that only in the G4 motifs we have this increase of error rate in motifs compared to controls. So in the stringently filtered set, that's still visible and actually a little bit increased. And then we also have this increase in error rate in the zDNA motifs. So overall, the picture is rather mixed, I'd say, but in general, G4 motifs show increased error rates across all 3 technologies. And this is fairly remarkable considering that these three technologies apply fundamentally different sequencing strategies or approaches. And also, while Illumina and PacBio share the use of DNA polymerases, Nanopore actually does not use polymerases. So this is quite interesting that still the result in the G4 motifs is consistent across all three. So we then ask ourselves, does this have any sort of real life implications? So what do we need to be careful about when we're actually studying these non-beam DNA motifs? And so for this, we developed a probabilistic approach to estimate the number of false positive single nucleotide variants using the error rates derived from the data we in the project. And so in this figure, there is the results of this. So on the x-axis of each panel, there is variable read depth going from 3 to 30x. And the y-axis shows the number, so it's logarithmic, and it shows the number of false positive single nucleotide variants. And for each panel, the three colors correspond to different allele frequency filters. So green corresponds to just a single genome, a single chromosome showing the variant, versus tripletons shows 3 chromosomes, and the turquoise color is then for a minor variant frequency of 0.01. So, and to summarize all these figures, where like the columns correspond to different numbers of individuals, ranging from 100 to 10,000 individuals. And rows, again, correspond to Illumina, HiFi, and ONT. But across all these technologies and sample size situations, it is pretty evident that the increase of read depth reduces the number of false positives. And for a typical study where you have 100 individuals, and you're interested in singletons and you use Illumina data, for example, from a read depth of about 20x, there is no false positive to be expected anymore. And yeah, so importantly here, the solid line denotes the non-B DNA, and the dashed line is the control sequence. So across all these, the controls, of course, show fewer false positives than the non-Bs. Okay, so this already brings me at the end of my talk. And to briefly summarize here, I would like to mention that there is ample evidence that non-B DNA is involved in a variety of very important cellular functions, but also diseases. The DNA polymerases and helicases applied in sequences might actually interact with non-B DNA structures and therefore cause some inaccuracies. And finally, the non-B DNA motifs seem to be associated with sequencing accuracy in a non-random way, and referring to the probabilistic estimation of false positives, I would say that is particularly in situations of low read depth, caution is advised. So this study will soon be published in Genome Research, but it is already available on bioRxiv for everyone who's interested in reading further. And with that, I would like to thank in particular my PI, Katerina Makova, and also Marcia Cremona, who is the co-first author, and of course, you for your attention. Thank you very much.